Welcome back to Studious and to one of the most structurally magnificent topics in the entire field of histology. Today, we are decoding bone. Not just the familiar white, rigid structure you picture, but the living, dynamic, constantly remodeling tissue that is one of the most metabolically active organs in your entire body. Yes, bone is an organ. On the left of our title slide, we see the familiar architectural blueprint of the human skeleton. On the right, we have a stunning histological micrograph showing the microscopic interior of bone. And already, we can see the vocabulary we'll be mastering. The mineralized bone matrix stains eosin pink. The osteoid seam, the unmineralized, newly laid bone, stains hematoxylin blue. Scattered throughout, we see hematopoietic cells, mesenchymal stem cells, osteoblasts, a Haversian canal at the center, and the delicate radiating tunnels called canaliculi. Here is what makes bone extraordinary. It is simultaneously the body's structural scaffold, its mineral reservoir for calcium and phosphate, and the birthplace of every blood cell you will ever produce. And it is perpetually being demolished and rebuilt every single day of your life. This video will take you through osteogenesis from the ground up, how bone forms, which cells build and destroy it, how it grows, and what happens when the process breaks down clinically. Let's build this together.
Osteogenesis, literally bone genesis, is the biological process of bone formation, and it is far more complex than simply depositing calcium into a scaffold. It is a highly coordinated series of cellular events: cell migration, proliferation, differentiation, matrix synthesis, and ultimately programmed cell degeneration, all working in concert. Critically, osteogenesis does not follow a single pathway. There are two entirely distinct routes by which bone can form in the body, and each is used in a specific anatomical context. Path 1 is intramembranous ossification. In this pathway, bone forms directly. Mesenchymal stem cells cluster together within a highly vascularized connective tissue environment and differentiate directly into bone-producing cells without any intermediate template. This is the pathway used to form the flat bones of the body: the skull, the mandible, parts of the clavicle, and the ribs. Path 2 is endochondral ossification. This is far more complex. Here, bone does not form directly. Instead, a pre-existing hyaline cartilage mold, shaped exactly like the future bone, is systematically broken down and replaced by ossified tissue. This is the pathway responsible for all of the long bones of the body: your femur, tibia, humerus, radius, and every phalanx. Understanding these two pathways is foundational because they explain not only normal skeletal development, but also why certain diseases, like achondroplasia, affect only specific bones. Keep that in mind as we proceed.
Let's now walk through the three-stage process of intramembranous ossification in detail. Stage 1 is the environment. The process begins within highly vascularized connective tissue that is densely populated with mesenchymal stem cells. These are multipotent progenitor cells, meaning they retain the capacity to differentiate into multiple cell types depending on the biochemical signals they receive. The rich blood supply is critical. It will deliver the calcium, phosphate, and oxygen needed to mineralize the future bone. Stage 2 is the osteoid. In response to local signaling molecules, primarily bone morphogenetic proteins, or BMPs, and Wnt pathway ligands, mesenchymal stem cells commit to the osteoblast lineage. These newly differentiated osteoblasts begin synthesizing a soft, unmineralized extracellular matrix called osteoid, composed primarily of type 1 collagen and proteoglycans. Under a light microscope, this fresh osteoid stains blue with hematoxylin, clearly visible at the osteoid border in our micrograph. The osteoid is laid down layer by layer, a process called appositional growth. Stage 3 is the final product. Once the osteoid reaches a critical threshold, osteoblasts secrete alkaline phosphatase,
an enzyme that hydrolyzes local inhibitors of mineralization and elevates the local concentration of inorganic phosphate. Hydroxyapatite crystals, calcium phosphate, begin nucleating within the collagen framework, hardening the matrix. Small islands of calcified bone, called trabeculae or bony islets, form first, then grow and fuse into a progressively more solid structure.
No bone formation story is complete without a thorough introduction to the two most important cellular players in intramembranous ossification: osteoblasts, the builders, and osteocytes, the managers. Osteoblasts arise from mesenchymal stem cells and represent the active bone-building phenotype. Their defining job is to synthesize and secrete the osteoid matrix, type 1 collagen combined with proteoglycans like osteocalcin and osteopontin, which form the organic scaffolding of bone. Their second critical function is the secretion of alkaline phosphatase, which drives calcification by mineralizing the osteoid. An important clinical correlate: serum alkaline phosphatase is elevated in conditions of high osteoblast activity, including Paget's disease of bone, bone metastases, and during fracture healing. It is a direct biochemical readout of how hard your osteoblasts are working. Osteocytes are the mature, post-mitotic descendants of osteoblasts that became physically entrapped within the mineralizing matrix they produced. Each osteocyte sits in a small cavity called a lacuna and extends long cytoplasmic processes through narrow tunnels called canaliculi, forming a network that resembles a neural web. Osteocytes are the mechanosensors of bone. They detect mechanical loading and strain and respond by secreting signaling molecules like sclerostin and prostaglandins to modulate osteoblast and osteoclast activity. They also facilitate the transfer of calcium and phosphate ions from the bone interior to active growth surfaces, contributing to mineral homeostasis.
For every builder, there must be a demolition crew, and in bone biology, that role belongs exclusively to the osteoclast. Osteoclasts are among the most distinctive cells in the entire body, and their origin is completely different from osteoblasts. They do not arise from mesenchymal stem cells. Instead, osteoclasts are formed by the fusion of monocytes and macrophages, cells of the hematopoietic lineage. This fusion process produces a large, multinucleated giant cell, typically containing 5 to 20 nuclei, with a characteristic pale, foamy cytoplasm visible under the light microscope. Osteoclasts work in 4 precisely coordinated steps. First, location. They seek out and occupy specific erosion pits on the bone surface, called Hauschip's lacunae, also known as resorption bays. Second, the anchor. Integrins on the osteoclast cell membrane bind tightly to fibronectin proteins on the bone surface, creating a hermetically sealed compartment between the cell's underside and the bone, isolating the resorption zone. Third, the ruffled border. The osteoclast develops an extensively folded basolateral membrane called the ruffled border, dramatically increasing its surface area for secretion and uptake. This is the functional business end of the osteoclast. Fourth, the payload. Vacuoles within the osteoclast fuse with the ruffled border membrane and release lysosomes packed with acid phosphatase and hydrogen ions, creating a highly acidic microenvironment that dissolves the hydroxyapatite mineral and degrades the collagen matrix. The liberated calcium and phosphate are then transcytosed across the osteoclast and released into the bloodstream. Clinically, bisphosphonate drugs used in osteoporosis treatment work by inducing osteoclast apoptosis, directly targeting this demolition mechanism.
Now we move to the second and more complex pathway, endochondral ossification, which is responsible for the formation of every long bone in your body. The defining feature of endochondral ossification is that bone never forms from scratch. It always replaces a pre-existing cartilage model. Let's follow the 3 stages. Stage 1, the cartilage mold. During intrauterine life, in approximately the 5th to 6th week of embryonic development, condensations of mesenchymal cells form and differentiate, but this time not into osteoblasts. They differentiate into chondrocytes, producing a hyaline cartilage mold that is shaped exactly like the future long bone. This cartilage model is avascular, flexible, and surrounded by a fibrous sheath called the perichondrium. Stage 2. Diaphysis mineralization. At the primary ossification center, located in the central shaft, or diaphysis, chondrocytes begin to hypertrophy. They enlarge dramatically, calcify their surrounding matrix, and then die. This calcification is driven by the same alkaline phosphatase mechanism we saw in intramembranous ossification. Simultaneously, blood vessels and osteogenic cells from the surrounding perichondrium invade the calcifying cartilage. Osteoblasts use the calcified cartilage scaffolding as a template, laying down osteoid around it. Stage 3. The bone collar. As the primary ossification center expands longitudinally, osteoblasts in the perichondrium form a structural bone collar around the diaphysis, a ring of compact cortical bone that physically constrains radial expansion and channels all growth longitudinally. The perichondrium at this stage earns a new name, the periosteum.
After birth, endochondral ossification does not stop. It continues at a specialized structure called the epiphyseal plate, also known as the growth plate or physis. This is the engine that drives the longitudinal growth of every long bone from birth until the end of puberty. The epiphyseal plate is situated between two critical landmarks: the epiphyseal calcification center at the very tip of the bone, the epiphysis, and the main central shaft below, called the diaphysis. It is a cartilaginous disc, and its cells are organized into 4 beautifully ordered columnar zones, each representing a distinct stage of chondrocyte maturation. The plate is regulated by a complex interplay of hormones. Growth hormone stimulates chondrocyte proliferation via IGF-1, thyroid hormone promotes maturation and calcification, sex hormones, estrogen and testosterone, initially accelerate growth before ultimately causing plate closure, and glucocorticoids, in excess, suppress chondrocyte proliferation and can cause growth retardation. At the end of puberty, typically ages 14 to 18 in females and 16 to 21 in males, rising levels of sex steroids cause the chondrocytes to cease proliferating. The cartilage is entirely replaced by bone, and the epiphyseal plate closes permanently, forming the epiphyseal line. Once this closure occurs, longitudinal growth is complete and irreversible. Clinical note: fractures through the epiphyseal plate in children, Salter-Harris fractures, are serious injuries because they risk damaging the growth plate and causing premature closure, resulting in limb length discrepancy.
Inside the epiphyseal plate, there is extraordinary microscopic organization. The chondrocytes are not randomly distributed. They are arranged into 4 distinct, color-coded functional zones, each representing a sequential stage in the life cycle of a cartilage cell. Understanding these zones is essential for both histology practicals and clinical reasoning. Zone 1 is the zone of reserve cartilage, also called resting cartilage. This is the uppermost zone, adjacent to the epiphysis. The cells here are small, irregularly arranged in random clusters, and have very low mitotic activity. Their role is not to divide actively, but to serve as a stem cell reservoir, maintaining a pool of progenitor chondrocytes that can be recruited into the proliferative zone below. Zone 2 is the zone of proliferation. Here, chondrocytes are actively dividing by regular mitosis and organize themselves into neat, parallel columns oriented along the long axis of the bone, like stacked coins. This is the primary driver of longitudinal bone elongation. This zone is most active during childhood and adolescent growth spurts. Zone 3 is the zone of maturation and hypertrophy. Cell division stops. Chondrocytes enlarge dramatically, up to 5 times their original size, accumulate intracellular lipids and glycogen, and begin secreting alkaline phosphatase. This enzymatic activity mineralizes the surrounding matrix, preparing it for vascular invasion. Zone 4 is the zone of calcified cartilage. The hypertrophied chondrocytes die by apoptosis. Blood vessels and mesenchymal osteogenic cells invade the vacant lacunae. These cells differentiate into osteoblasts and lay down osteoid on the calcified cartilage framework, converting the template into true bone. This is the zone immediately adjacent to the diaphysis.
Between the mineralized trabeculae of bone lies a tissue that is just as important as the bone itself: Bone marrow. The space between the bony trabeculae is not empty. It accommodates a highly vascularized, metabolically active hematopoietic environment. When you look at a bone marrow histological section, you need to be able to identify 4 key components simultaneously. First, the collagen trabeculae. These are the rigid, pink, mineralized structural borders that define and partition the marrow space. They are the bony framework, the walls of the marrow compartment, appearing as dense, eosinophilic beams and struts under low magnification. Second, the highly vascularized background. Threading between the trabeculae is a high density of blood vessels, sinusoids, and arterioles. This rich blood supply delivers nutrients, growth factors, and hormonal signals, while also providing the exit route for newly produced blood cells entering the circulation. Third, the voided areas. Scattered throughout the section are empty white circular spaces. These are lipid-filled fat cells, adipocytes, whose fat dissolved during tissue processing, leaving behind ghost-like lacunae. The ratio of fat to hematopoietic tissue changes dramatically with age. In newborns, virtually all marrow is red, active and hematopoietic. By adulthood, the appendicular skeleton has largely converted to yellow marrow, fat-filled and relatively inactive. Fourth, cell-rich niches. Dense clusters of small, darkly staining hematopoietic cells represent active blood cell production. Erythroid precursors, myeloid precursors, and megakaryocytes, all visible in a single section.
We have now covered both pathways in depth. Let's crystallize the essential comparative knowledge into a clean diagnostic framework, because in your exam, you must be able to distinguish intramembranous from endochondral ossification instantly. Compare them across 4 dimensions. Starting with the initial environment, intramembranous ossification begins in highly vascularized connective tissue, rich in mesenchymal stem cells. Endochondral ossification begins with a preformed hyaline cartilage mold. Cellular mechanism, intramembranous uses direct mesenchymal to osteoblast differentiation. No intermediate cell type required. Endochondral is mediated entirely by chondrocyte hypertrophy and death. Cartilage must be produced and then systematically eliminated before bone can form. Growth pattern. Intramembranous produces appositional growth. Bone is laid down layer by layer in concentric sheets, thickening outward. Endochondral produces longitudinal growth, driven by chondrocyte proliferation in the columnar zones of the epiphyseal plate, pushing the bone ends further apart. Bones formed, intramembranous ossification produces the flat bones, the skull vault, mandible, clavicle, and parts of the facial skeleton. Endochondral ossification produces all long bones, femur, tibia, fibula, humerus, radius, ulna, and the phalanges. Memory anchor? Flat bones equal intramembranous. Long bones equal endochondral. The mechanism matches the mechanical demand. Flat bones need area, so they grow by apposition. Long bones need length, so they grow by longitudinal endochondral replacement.
Now we connect the cellular machinery to real disease. And the first case study is Rickets, one of the most historically significant and globally prevalent childhood bone diseases. Rickets is fundamentally a failure of osteoid mineralization in children, caused primarily by vitamin D deficiency. To understand why, you need to understand what vitamin D normally does in bone metabolism. In the normal cycle, osteoclasts resorb existing mineralized bone, liberating calcium from the matrix into the bloodstream. This freed calcium becomes bioavailable. Simultaneously, osteoblasts lay down new osteoid. Vitamin D, specifically its active form, 1,25-dihydroxycholecalciferol, is essential for promoting intestinal calcium absorption and for stimulating osteoblast mineralization. It closes the loop. Without it, the calcium freed by osteoclasts is not replenished from dietary intake, and the new osteoid laid down by osteoblasts cannot mineralize. The result? Osteoblasts keep building osteoid, but the osteoid remains permanently soft and unmineralized, like wet concrete that never sets. When a child bears weight on unmineralized bone, the bone lacks compressive strength and buckles under gravity. The classic clinical picture emerges.
bilateral bowing of the lower limbs, often forming a characteristic 60-degree angle, swelling of the wrists and costochondral junctions producing the rachitic rosary, frontal bossing of the skull, and delayed tooth eruption. Treatment with vitamin D supplementation reverses the mineralization defect rapidly, a testament to the direct causal link between a single molecular deficiency and macroscopic skeletal deformity.
Our second clinical case strikes at the very heart of endochondral ossification, and it is the most common form of short-limb dwarfism in the world, achondroplasia. Achondroplasia is caused by a gain-of-function mutation in the FGFR3 gene, the fibroblast growth factor receptor 3. In the normal state, FGFR3 signaling acts as a brake on chondrocyte proliferation, preventing runaway cartilage expansion. In achondroplasia, the mutation makes the receptor constitutively active. It signals continuously, without needing a ligand to activate it. This means chondrocyte proliferation in the zone of proliferation of the epiphyseal plate is drastically and permanently suppressed. The consequence is selective and precise. Endochondral ossification, which depends entirely on normal chondrocyte proliferation in the epiphyseal plate, is severely impaired. The long bones of the limbs, femur, humerus, tibia, radius, fail to achieve normal longitudinal growth, producing the characteristic short proximal limb shortening called rhizomelia. But here is the key insight that makes achondroplasia such a powerful teaching case. Intramembranous ossification is completely unaffected. Because the skull, mandible, and ribs grow by direct mesenchymal ossification, no chondrocytes, no cartilage mold, no FGFR3 dependence, they develop with entirely normal dimensions. This explains the characteristic phenotype of achondroplasia: very short limbs alongside a normal-sized trunk and skull, with a relatively prominent forehead from compensatory cranial base changes. In 2021, vosoritide, a CNP analog that suppresses FGFR3 signaling, became the first approved drug treatment for achondroplasia in children, directly targeting the molecular defect.
We close by consolidating everything into your 4 essential frameworks, the foundation for any exam question on bone histology and osteogenesis. Framework 1, the 2 blueprints. Bone forms by 2 pathways. Intramembranous takes vascularized connective tissue directly to flat bones via mesenchymal to osteoblast differentiation. Endochondral takes a hyaline cartilage mold to long bones via chondrocyte hypertrophy, death, and replacement. Framework 2, the cellular triad. 3 cell types govern all of bone biology. Osteoblasts build. They secrete type 1 collagen osteoid and express alkaline phosphatase to mineralize it. Osteocytes manage. They sense mechanical loading from within their lacunae and orchestrate adaptive remodeling responses. Osteoclasts demolish. They use acid phosphatase and hydrogen ions to dissolve mineralized matrix, liberating calcium for systemic use. The balance between osteoblast and osteoclast activity determines whether bone mass is gained, maintained, or lost. Framework 3, the growth engine. Longitudinal bone elongation is entirely driven by the 4-zone epiphyseal plate: reserve, proliferation, maturation, and calcification. It remains active until puberty closes it permanently. Framework 4, the 2 points of failure. Rickets disrupts the raw materials. Vitamin D deficiency means osteoid cannot mineralize. Achondroplasia disrupts the instructions. FGFR3 gain-of-function mutation means chondrocytes cannot proliferate. One hits mineralization, the other hits the cellular signal. Both produce devastating skeletal consequences, and both are explained entirely by the molecular mechanisms we covered today. Thank you for watching Studious. If this atlas helped you, subscribe, leave a comment with your questions, and share it with a fellow med student who needs it.